Alright guys, so this is going to be our last video on Punnett squares, and after we do this you should be able to hopefully go through the rest of your dragon packet and finish up all of your genetics problems. So we have two more that we need to look at that are coming from the notes that you did. We have blood typing problems and sex-linked Punnett squares. We're also going to take just a quick look at pedigrees, and we'll take a look at a couple other examples that are in your dragon packet. And so we're going to start with blood typing. And so a lot of what we have here, if you're looking in your notes, you actually have a lot of it right there to look at. You don't necessarily have to write all this down from scratch. But I just wanted to go over a few things because there are some, vocabulary issues. So first off, when it comes to your blood typing, we have, for ABO blood group, we have three alleles: the A allele, the B allele, and the O allele. Now remember that another way to represent those are with the I's. Now the I comes from the word immunoglobulin, which is a protein that's on your blood cells. It doesn't matter to me which ones that you use. The main thing is you should recognize these as those alleles. Okay, so you can use either method. You should come out with the same answers either way. I personally just like to use A, B, and O because I know that these are both dominant and this is recessive. Okay, so you can use whatever you feel more comfortable with, but you should at least recognize the I's if you ever see those. Now, these three alleles can produce 6 possible genotypes. Okay, this is one of those examples of multiple alleles, and so we can see we have a greater variety of genotypes and phenotypes. Now, the genotypes, as always, are your letter combinations. You could use the I's if you prefer, but these are my 6 possible combinations, and the genotypes determine the phenotypes. In the case of blood typing, the phenotype is your blood type. You are type A, you are type B, you are type AB, or you are type O. Now, one thing I want to be clear on is if the question asks for a blood type, these are not my blood types. These are my genotypes. Nobody says I'm AA positive or I'm AO, okay? You're type A. So the genotypes are your 2-letter combos, and the phenotype is the actual blood type. And the only one of those that has 2 letters is your AB because of their codominance. And so we're going to take a look at a couple of problems in your practice packet. We've got the heterozygous type A mom. We're looking at numbers 1 and number 2 in your practice packet. So number 1, it says a heterozygous type A mom. So the same terms apply as what we've seen before. So if it's type A, we have 2 possibilities. She's either AA or AO, but it tells us that she is heterozygous. So we know that mom is AO. Okay, everything we need is going to be in the problem. And she has children with a man that is type AB. So the only way to get AB is if you have an A allele and a B allele. So there's dad. Inside we represent our possible offspring. I do always write O second because O is recessive. Okay, you could have the I's in there, that would be completely fine. And so now we answer the question. So we're kind of following those same basic 5 steps on a problem like this. What are the possible blood types? Now this is important because it says blood types. It doesn't say genotypes. So what are the possible blood types? So we're looking for phenotypes. So here we have an AA and an AO. So those children would be type A. We have an AB. And we have a BO, which would be type B. So this is the answer to my question. The possible blood types of my offspring are A, AB, or B. Okay? Now the next question is a little bit different. A lot of times in blood typing we have to make some assumptions. This is kind of like one of those problems where we're working backwards, where they're saying, hey, could this be the parent? So normally the mom isn't in question. Sometimes you could see problems where, was the baby switched at the hospital or something like that. But normally it's the dad that's in question. And the thing with blood typing is all we can do is say, yes, they could be the parent, or no, they could not. Just because someone has type B blood and a dad has type B doesn't make them the father, because there's millions of people that have type B. So all we're doing with this is we do have to make some assumptions sometimes, but when it comes to blood typing, all we can do is kind of rule out or we could say, yes, it is a possibility. And that's kind of what this last problem is. So it says a child has type O, okay? So the only way that a child could be type O is if their genotype was OO. So this one is not following our typical 5 steps, okay? So our child is OO, and it says her mom is type B. Okay, now this is where you have to make some assumptions. Now mom has 2 possibilities. If she's type B, she's either BB or BO. Now when we look at her kid, we know that the mom had to have given that child an O. Okay, so we know that mom is BO. So even though they didn't tell us what her genotype is, we can make an assumption based on her child, okay? And so the question is asking us, we have a man who is type A, can he be the father? And so in a problem like this, you may or may not have to do a Punnett square, okay? You can show your work or kind of prove it with the Punnett square. So we know that mom was BO. Now if dad was type A, his possibilities are AA or AO. Now if he's AA, he's not going to have that child. But if he is AO, there is a possibility that that combination of parents could produce an O child. So on a problem like this, we would simply say, yes, it is possible. It may not be him, but yes, it is possible if he has an AO genotype. Okay, so when you're doing blood typing, that's how you're going to go about doing it. Now, in your dragon packet, you do have a dihybrid cross that's looking at both components of blood typing. So one component is the ABO blood group. The other component is the RH factor, okay? So when you're doing the RH factor, that is just a separate gene. This is just like doing any other dihybrid. You have 2 traits. You have ABO and you have positive negative. Now, when you go to work that dihybrid, you can do it a couple of ways, okay? As always, I would make a key. I'm going to use R for RH, but you could use any letter of the alphabet. A lot of times the letter D is used because it's the D antigen. But we have a dominant trait and we have a recessive trait. So when you're doing positive and negative blood, it is a completely separate gene, completely separate key, okay? Now being positive is dominant and being negative is recessive. So you're going to work those out just like a normal Mendelian problem. So you could have a homozygous positive person, they would have 2 capital R's. You could have a heterozygous person with positive blood, capital R, lowercase r. Or you could have a person with negative blood, and that would be little r. So in your practice, where you have to do a dihybrid, you're just combining the ABO blood group and the RH blood group. So just like any other dihybrid, you're going to have 4 letters in your parent genotypes. So for example, let's say I had a heterozygous type A positive blood type. Okay, a person is heterozygous for type A and they're heterozygous positive. So I would write that person's genotype AO, big R, little r. Okay, so it's the same way we did the other dihybrids. You would foil this, you would set up your dihybrid. So if you have any questions about that, you can maybe go back and watch this part of the video. So we don't have any just regular monohybrid positive negative problems, but that's the key for it, okay? Positive is dominant, negative is recessive. All right, the next thing we're going to look at is sex linkage. Okay, so remember that you have 44 autosomes. That's chromosome pairs 1 through 22. And on your autosomes, everybody's getting 2 copies. All of these traits where we just wrote 2 letters in our genotype, those are all autosomal traits. We didn't have to look and try to decide, you know, we didn't have to think about whether they were male or female or anything like that. All we had to do was just write the letters. They were homozygous dominant, heterozygous, or homozygous recessive. Now, if it's a sex-linked trait, it's a little bit different because this means that it is on the chromosomes on pair 23. It's either going to be on the X chromosome, and we call that an X-linked trait, or it's going to be on the Y chromosome, and we call that a Y-linked trait. Now, as I mentioned in our notes, the Y-linked traits are not very common because there's very few genes on that little tiny Y. So normally when we're talking about sex-linked, we're talking about X-linked, all right? So the difference here is that boys and girls are going to get a different number of alleles because they're either getting XX for a girl or they're getting XY for a boy. So where a girl gets 2 copies of each of those genes, the boys only get 1. And so that will lead to a difference in inheritance among boys and girls. So we have to include the sex of the individual as part of their phenotype. We also have to include the sex of the individual in their genotype as well. So there's a section right under blood typing. Okay, you have an example. And so the example that I gave you was for color blindness. Okay, so red-green color blindness is an X-linked recessive trait. Now, I would highly encourage you to make a key for your X-linked traits, just like you would any other type of trait, okay? So it tells us that color blindness is an X-linked recessive trait. So the little b is going to represent color blindness. The capital B, the dominant trait, is normal vision. Once again, the key will always help you avoid mistakes, okay? So here's my basic inheritance. I have dominant as normal vision, recessive is colorblind. Now, in this case, when we write our genotypes, these alleles are on the X chromosome. Okay, so we're going to have to include our XX and our XY as part of our genotype. Because once again, girls are going to get 2 copies, boys are only going to get 1. And so we write the alleles as superscripts on the X. So for girls, there's 3 possible genotypes. They could get homozygous dominant, they could be heterozygous. Now, because it's a recessive trait, this is what we would call a carrier, all right? She carries that recessive allele, but she is not colorblind. A carrier does not have that recessive trait, okay? She carries the allele, but she does not have the trait. In a sex-linked trait, only girls can be carriers. If it's an autosomal trait, anybody can be a carrier, okay? Now, the third possibility for a girl is she could be homozygous recessive. Now, for boys, there's only 2 options because they get a Y instead of a second X. They only get 1 copy. So they either get a big B or they get a little b, all right? Now, when it comes to your phenotypes, we have to include the sex of the individual as part of the phenotype. So like this girl would be dominant. So in this trait, that would be a normal vision girl. This would be a dominant girl, heterozygous. Okay, she's normal. The only way a girl could be colorblind is if she got 2 recessive alleles. And then for the boy, it's a 50-50. They're either normal or they're colorblind, okay? And I wrote that wrong, I should say boy. Little mistake there. So now we're going to work out our Punnett squares for sex linkage. As always, we're going back to our 5 basic steps. Okay, so we're going to take a look at our 2 examples that you have here. Number 3 says a carrier female. Okay, now we're using the key that's up above these problems, the one that we just had written on the paper that I just ripped off. So a carrier female, so if it's a female, we know she has 2 X's, and carrier means she is heterozygous. So she's going to have a big B and a little b. All right, we have to write all of those components of the genotype. She's married to a man with normal vision, so we know that a man is XY. Now, if he's normal vision, he has a capital B. He only gets 1 B, okay? Nothing goes with the Y. The Y is just Y. Now, this right here is also why you have a 50-50 chance of having a boy or a girl, because the boy either gives an X to the baby, and it's a girl, or the boy gives a Y to the baby, and it's a boy. So it's actually the male that determines, I mean, not by choice, but it's the sperm that determines whether that baby is male or female. So inside, as always, these represent our offspring, and so we're just going to put whatever's above and beside the box. So our top 2 offspring are going to be girls. This girl has 2 big B's. This girl is a carrier, okay? The bottom 2 children are going to be boys. Remember, the Y is just a Y. This boy got a capital B. This boy got a lowercase b, okay? So the boys are inheriting this trait from their mother, okay? The girls are getting the X's from both mom and dad. And so now that we have our problem complete, now we just have to answer the questions. Now there is a slight difference in questions sometimes with sex linked. So the first question says, what % of their children would you expect to be colorblind? So if we're looking at all of our children, this girl has normal vision, this girl has normal vision, this boy has normal vision, but he's colorblind. So for children, 25% are colorblind, because 1 out of 4. Now, if it asks about individual genders, like how many boys or how many girls, it's no longer out of 4. I've got 2 possible girls and I've got 2 possible boys. So if we're talking about male or female, our percentages are going to be 0, 50, or 100. So the second part of the question says, what percent of boys? So out of my boys, half of them would be colorblind. And then the other question says, how many girls, or what percent of girls? Well, both of our girls have normal vision. Remember that carriers do not have the recessive trait, they just carry that allele, so they're going to have normal vision. So 0% of my girls are going to be colorblind. So you just need to pay attention to the question. Are they asking about children as a whole, or they're asking about males or females? All right, so let's do one more practice problem, number 4. A homozygous normal vision female. Okay, so as always, a female is XX, and it says she's homozygous normal, so that's going to be 2 capital letters. Okay, she's crossed, or she mates with a colorblind male, so male is XY. If he's colorblind, that means he has the lowercase b. Remember, it's important that you probably have that key. We wrote it on the page before, but we should have a key that says big B is normal and little b is colorblind. So as always, we perform our cross. All right, I get 2 girls and 2 boys. And now we answer the question. What percent of their sons would you expect to be colorblind? So when we look at our boys, okay, they both have capital B's. And so 0% of our boys would be colorblind. In fact, none of our children are going to be colorblind. If that mom is homozygous dominant, everybody's getting a capital B. No one is going to be colorblind in those children. Okay, so I have 2 carrier females, and then I have 2 normal vision boys. Now, this may also explain on pedigrees how we see that girls are the only ones that can be carriers. If you were to go back to the lesson and look at that color blindness pedigree, remember, only a girl can be a carrier because a carrier is a heterozygote. A boy only gets 1 copy. So boys cannot be carriers in a sex-linked trait, okay? Boys can be carriers if it's autosomal, but they can't if it is sex-linked. All right, so that's going to wrap up the genetics problems, but I do want to look at a few more things in your dragon packet. So I hope you have those handy. The first thing I want you to look at is there's a page that has some problems. We're not going to do them together, okay? I just want you to take a look. It actually explains pretty thoroughly how you do these. These will not be on your quiz or your test, okay? So these types of problems are not going to be on your quiz or your test. But I did want you to see that there's some other types of inheritance. So one example are lethal genes, and this is where some genes, when they're inherited together, they lead to incomplete development. So one example, there's actually 2 examples in humans. One is Huntington's disease. It's a dominant disorder, but if a child gets 2 dominant alleles, they usually don't complete development, so they don't make it. The same thing occurs in achondroplasia, which is a type of dwarfism. So if they get double dominant, it's a dominant trait, but double dominant usually doesn't survive very long. So if you see someone, like an adult with dwarfism, the achondroplasia, a specific type, you know that they're heterozygous, which is why you can have 2 people with dwarfism having normal height children, okay? So that's just an example. So what pretty much what happens is when you get that double, you kind of X out that offspring, and so now instead of out of 4, you have 3 surviving offspring. So there's an example there with dwarfism. Manx cats, where they have like chopped off tails, that's another example of a lethal gene. There's a certain kind of chicken that has a lethal gene. I can't remember exactly, it's the legs that kind of curve backwards. I can't remember what it's called. So there's a few different examples of lethal genes that you could come across. Once again, not on the test. The other example is epistasis or epistatic alleles. And so this is where you can have two different genes and one affects the expression of the other. So you see this a lot in pigmentation, okay? And one example is like what you have here in labs. If you're familiar with dogs, there's pretty much three colors in your labs. You've got the black labs, the chocolate labs, and the yellow labs. So the way epistasis works is one gene determines the color of the lab. The lab is either supposed to be black or they're supposed to be chocolate, okay? Black is dominant to brown. But there's a whole separate gene that determines whether or not that dog has the pigment. So it's dominant to have the pigment. It's recessive to not have the pigment. And that's essentially how you end up with a yellow lab. So on one gene, it could say that they're brown or black. But if on this other gene it says that they don't have the pigment, they're going to be yellow, okay? So the pigment gene kind of overrides the actual color gene. If they don't have pigment, it doesn't matter what color they're supposed to be, they're going to be a yellow lab. And so I've given you a key that kind of shows you all the genotypes, okay? You're just working on a dihybrid cross. The only difference is you have an additional phenotype because of that expression. So epistatic alleles are when the expression of one gene can affect the expression of another. So I want you to do the problem, I want you to be familiar with it, but it's not going to be on the test or quiz. All right, the last thing we're going to look at are pedigrees. Now remember that these are like genetic family trees. I'm going to do one more paper. It's going to be too hard to see it on the little paper. So with pedigrees, remember that we are tracing a trait. We can trace any trait that exists. We can trace a dominant trait, we can trace a recessive trait, we can trace autosomal, sex-linked. It doesn't matter what it is. It could be a disease, it could be something just completely normal like freckles. You just trace a trait. It doesn't matter what it is, okay? Now, in looking at pedigrees, we can kind of answer questions and look at inheritance and possibly look at genotypes, okay? So if we look at the very first example, I'm going to quickly draw this. All right, so notice this is generation number 1. It has a Roman numeral 1. All right, so they have 3 kids, got a circle, got a square. She married. And we have all right, so here's a pedigree that you have on your paper. Okay, we're going to answer questions 1, 2, and 3 about this pedigree. So this is generation 1, this is generation 2, and this is generation 3. And notice they're numbered. So this is like individual 1, 1, 1, 2. Okay, so that's how you kind of identify the people in a pedigree. Remember that circles are females and squares are males. If they're filled in, they have whatever the trait is. If they're not filled in, they do not have the trait. Sometimes you will see half filled in, and that means they're a carrier. But we only see carriers if the trait is recessive. Okay? So let's take a look at the questions that you can answer. So number 1 says, how many males in this family have the trait being traced? So first off, you have to know that males are squares. Okay, so we're only looking for squares. And if they have the trait, they're filled in. So as we look through, out of this whole family, there's 1 male that has the trait being traced. Okay. Second question, how many females have the trait? So now we're looking for circles that are filled in. There's 1 and there's 2. And then the last question says, if the trait is autosomal dominant. Now what that means is it's on chromosome pairs 1 through 22. Everybody's getting 2 copies. Don't have to worry about male versus female, okay? Males and females are on an equal playing field when it's autosomal dominant. And in fact, if you have an autosomal trait, you should not see a significant difference between male and female. I would not consider 2 to 1 in a small pedigree like this a significant difference. So they're telling us that it's a dominant trait. And so what they're asking for is the genotype of individual 2, 2. All right, so we're looking for this individual right here, okay? We want to know the genotype. Now, because it's autosomal, our genotype is just going to have normal O letters. 2 letters, either capital, 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 lowercase. If it's not sex linked, we do not include the XX and the XY, okay? So we have a female, that really doesn't matter. All that matters is that she has the trait. So if it's a dominant trait, she must have a capital letter, okay? So we know that she's either capital A, or capital A, lowercase a. Now, can we determine which one she is? Sometimes we can, sometimes we can't. In this one, we can, okay? And so the way we can determine what her genotype is we can look at her family. So we know she has at least one capital A. But if we look at her family, up here, her father is not filled in. If they're not filled in a dominant trait, that means they're recessive. So her dad is little a, okay? So if her dad is recessive, then she must have gotten a little a from her dad. And so for this particular pedigree, she must be a capital A, lowercase a. We know that she's heterozygous because she has the dominant trait, but her dad doesn't. Okay, so a lot of times you can look at parents or you can look at kids. Now, if you look at number 4 and 5, we have a new pedigree there. The first question says, is the trait most likely autosomal or sex-linked? Once again, what you're looking for there is if it's autosomal, you should not see a significant difference between male and female. If it's sex-linked, you're probably going to see more males that have it, only female carriers. Okay, you're definitely going to see some differences between male and female inheritance. And then once again, it's asking for genotype, and so you can kind of determine that from the information that's given. On the back, you have some other pedigree questions, okay? Like this one's asking, is it dominant or recessive? There's a very important clue on this one in that you have carriers, okay? So that's going to tell you whether it's dominant or recessive. Once again, autosomal or sex-linked has to do with do you have an even distribution among males and females? Autosomal traits are inherited the same among male and females. There's no difference, okay? Now, if you determine that it's autosomal, your genotypes are just normal letter combinations. If you determine that it is sex-linked, you have to include the sex of the individual. So if it's a female, you would have to include XX with the appropriate letters. If it's a male, you would include XY with the appropriate letter on the X. So that's how you go about doing your pedigrees, okay? I think at this point, we've covered everything that we need to cover. So you're going to have part of today maybe to work on your Punnett squares. You may have already done them. You're going to have a whole day tomorrow to do nothing but work on your Punnett squares. And so on your next quiz, it's going to be over human heredity, and it's also going to include all of the new problems that we've done, okay? You're going to have a dihybrids, you could have blood typing, you could have sex-linked, you could have the other problems where you like multiply the fractions or you figure out what the parents are from the babies. So be prepared for any of those on the quiz. Remember that lethal genes and epistatic alleles will not be on the quiz or the test, okay? But I still want you to do that in the practice packet. You're going to turn in that Dragon genetics packet the day that you take the genetics test. So you have until then to get it complete, but I would highly encourage you to work on it and try to get it complete so that if you have any questions while you're in class, you can get those answered. All right, that's all I have for you today. Y'all have a good one, and good luck on all of your Punnett squares.